Welcome to Science in the Virtual Pub. I'm Don Haas, your host for Science in the Virtual Pub. And I'm going to go back to our coming attractions. There we go. So there's where you can donate to support Science in the Virtual Pub. And tonight's session is Visualizing Science Through Art with Marla Coppolino. And we'll come back to her as I hit what else is coming up. This Saturday, if you're in Central New York, come on down to the Climate Education Resource Fair and Professional Development Workshop at the Science Center in Ithaca. That's 9 a.m. to noon on Saturday. We'll have 18 groups tabling there. 2 weeks from tonight in Science in the Virtual Pub, we'll have Rebecca Rundell from SUNY ESF talking about island and land sails, Paradise and America, or island land snails, I should say. And on October 11th, we'll have Teacher Resource Day at the Museum of the Earth in Ithaca. And again, all of these coming attractions that if you want to learn more about, you'll be able to check out that info in our newsletter. And on October 23rd, we'll have Dr. Suzanne Macy from the American Museum of Natural History talking about we need to conserve biodiversity. Improving our teaching will help. On October 27th, not a Science in the Virtual Pub event, but an event online, favorite climate lessons panel with some teachers who teach climate change electives, talking about their favorite lessons. And that's a part two of a session that we had in the spring. Then on November 13th, we'll have Dr. Saul Gruner from an emeritus physics professor from Cornell University, upending assumptions about life on Earth. And on December 11th, we will have our 100th episode special panel with previous guests talking about how their science has changed in the last 5 years. And at the Museum of the Earth and online, we've got our marvelous mollusks exhibit, and that is open now. I should update my graphic. That's left over from the spring, and you can, like I said, check out all of that stuff on our Learning Earth newsletter, which you can find there, and I'll put a link in the chat in just a moment. Oh, and I see that we've got people in the waiting room. A little bit of difficulty with me being solo here. Sorry about that. There we go. So here come 5 more people. All righty. I think I'm going to hand it off to Marla for talking about visualizing science through art. So I'm going to stop my share and take it away, Marla.
All right. Thank you for the introduction, Don. I'm Marla Coppolino. I wear many hats in my day job and as well as other parts of life. So I am going to bring up my slides. So today we're here to talk about visualizing science through art. And what does that mean? So for a long time,
We're not seeing your slides yet.
Oh,
I think you didn't hit the share button or didn't hit the follow up to the share button.
that's very important.
Yes.
How about now?
Now we see them. Thank you.
I'm sure are a lot of intermediate buttons to push, aren't there?
Mm,
Everybody see my slides?
yes.
Excellent. So, okay, let me see if I can collapse the sidebar so I can see what I'm looking at. Great. All right. For a very long time, we called science communication, we referred to it as science illustration. And I still sometimes oscillate between the terms science illustration and visual science communication. But what happened is in recent years, we decided that visual science communication, it's really a broad, more inclusive field that is not just about illustration, but It includes animation and sculpture and murals and digital and traditional illustrations and more, even interpretive dance, from what I understand. But I have no interpretive dance to show you this evening. I hope you're kicking back with a drink of your choice, and we'll look at some examples of science through art over time. I do want to say that another aspect of this definition is that it's a powerful communication tool. It plays a role in conveying science information to all. So scientists talk to scientists, and sometimes as a scientist, we forget and we might use jargon or certain phrases, and we miss some of our public audience. It's for many audiences, but we must be clear as visual science communicators who we're talking to. And we're talking to a broad audience today, so I'm going to keep this nice and broad. And I'm happy to answer questions at the end of my presentation. First, I want to give a quick shout out to my professional organization. Uh-oh. There. It's called the Guild of Natural Science Illustrators. It started in 1968. It was formed by a group of science illustrators in the Washington, D.C. area. And as soon as I learned about it in 1992, I joined. I had taken my very first university course in science illustration, and I was hooked because when I was a kid, I couldn't decide if I wanted to be an artist or a scientist. And it was presented to me that, hey, there's both. This can come together and you can use both sides of your mind and your interests to do this. Here locally in the Ithaca, Central New York area, we have the Finger Lakes chapter, and we've been a very active and eager group of local artists and illustrators here. And you're welcome to look into both of these. All right. Let me start by saying that our history as human beings, our species goes back a very long time as being creatures of habit of wanting to present a picture for somebody else to see. So way back in, let's see, how long ago was this? You've all probably seen this horse in the Lascaux cave in France. And it was believed to have been painted between 17 and 22,000 years ago. Think about that. That's a long time ago that humans were producing art. And what was it about? Um, as innately visual creatures, these people were painting animals and hunting scenes of their daily life. They themselves were probably just trying to process their own experience and express them to others and to remember them and have a record of them for others to see and experience through their artwork. We go back even further because in recent years we're learning more, and the Chauveau Cave, also in France, has numerous hundreds and hundreds of animal paintings on its walls. This is a painting of lions. Look how beautifully the heads are portrayed. Um, and this was even longer ago, 32 to 37,000 years ago. This was the visual communication. I think we can go back further. We can, because here, 45,000 years ago, archaeologists have found in Sulawesi in Indonesia some rock paintings that depicted pigs and lots of other animals. And they were also their prey that they hunted. And there's a lot of images of the pigs surrounded by humans. There's human figures in them as well. So let's just think about these images we've seen for a moment, right? Okay, sit with this. All of these paintings were made thousands of years before the earliest forms of written communication. This is what it was for most of the human experience. So when was written communication developed? Around 6,000 years ago in a form that was agreed upon and made tangible on clay tablets and such. That is just a mere drop of time in the greater scheme of things, including our own experience as humans on this planet Earth. So, to sum up, most of our time as a species, we've been communicating our experiences through pictures for others to see. So I'm going to bring you a little closer to modern times and just walk through a few of the notable artists who were considered visual science communicators. Everybody knows Leonardo da Vinci. Of course, he was an architect, he was an artist, he was a scientist, and he very much a Renaissance man and an engineer and architect as well. And when you look at his drawings, what do you see? What do you feel? I don't know about you, but I just get this overwhelming sense that his curiosity was insatiable. He wanted to, just needed to examine and dissect and understand and then draw the components of what he was looking at and how they fit together. The body of work that Da Vinci left us is enduring. It stands to this day, and this was a true visual science communicator. Going forward, maybe not as well known, but if you don't know her, she's worth looking into and learning more about. Maria Sibylla Merian was a German illustrator and scientist, and her favorite topic of study was caterpillars. And she examined and drew caterpillars and their life cycles. And what's especially notable about her work is that prior to what she did, people thought that insects were what they say born of the mud, and they reproduced by spontaneous generation, or not even reproduced, they were just born out of the mud, and they were believed to be evil creatures of the devil, but she saw them in the beauty that they truly are, and she captured all this in a series of she had published and illustrated a 2-volume series on caterpillars, as well as the plant that supported them. So the host plants for the caterpillars, she also studied and noted the associations between them. Next is Ernst Haeckel, a German, many things. A zoologist, he was a philosopher, a naturalist, also a physician and a professor. And he studied marine biology and particularly invertebrate animals, that's animals without backbones. He discovered and named thousands of new species that were unknown to science at that time. And he also had a good understanding of how life was related, and he would map out those relationships. He also left us many terms in biology. He coined terms that we use today, including ecology, phylum, phylogeny, ontogeny, and protista are all words from Ernst Haeckel. He also had a wonderful geometric and visually aesthetically pleasing way of depicting the organism. So they're truly works of art. Even if you don't know what they are, they stand as art that is beautiful to look at, but also informative and true to what these animals look like. Nice, we have Beatrice Potter, and I think all of you know her and probably appreciate her as being the English writer who wrote Peter Rabbit and all the wonderful books about animals. But Beatrix Potter was also a scientist, and she had a scientific mind. She wanted to know anatomy and compare different insects and fish to one another. She was especially accomplished in mushrooms and fungi. And she worked with scientists, and she herself published some work on mushrooms and mushroom spores. Hmm. And the last one I'm taking you on this brief tour of are the Blaschka father and son team, Leopold and Rudolph, and they were skilled glass blowers. They studied marine invertebrates for the most part and created their forms as true to life as possible because they saw them after they were captured on fishing boats and such. And for those of you who know, a lot of these organisms just fall apart and turn to mush immediately after you bring them up in a net from the sea, especially the one in the upper right, which is a type of sea slug. They just go to mush, they lose their color, and they don't look like this. And after a few minutes of being out of the water. So they created these glass blown models, and these became teaching tools especially for universities. So Cornell University in the early part of the 20th century purchased the full set of over 400 models for their biology laboratory classes for students to learn what these organisms look like in life. So it's very easy to look at these and get lost in their beauty and their beauty of being blown glass, beautiful objects. But remember, they were intended as teaching tools and a form of visual science communication. Okay, in this next section of my talk, I'm going to tell you all about the most common question I get. The number one question is, why not use a photograph? We live in the digital age of photography and just the digital age. And are we, as science illustrators and visual science communicators who don't particularly do photography, still relevant? Well, we are, and I'm going to show you why in the next series of slides. And at the same time, I give a nod to digital photography. I certainly use it, and it's very important. It's improved, and it has many uses that make it relevant today as well. But let's first go through some of these. So, first of all, if I didn't say so already, my area of study is mollusks, in particular land snails. Land snails are soft-bodied animals with a shell, and their structure, their anatomy does not hold up well if they are used for preservation and dissection. Even with fixing techniques, they're kind of mushy and they don't hold up like this. So the purpose of illustration for this was to show very clearly that structure at the top is actually the eye of the snail. It's on the upper tentacles, and I just wanted to illustrate some of their other organs and systems inside. So, if I didn't, the photographs don't work quite well for these. The specimens lose color, they might dry out, even if preserved, they're not ideal to learn from. At least not for the masses who aren't taking the time to sit over them for 8 hours a day and poke around and figure out what they're made of, right? Cross sections are another wonderful reason, an important reason we use um, illustration. Okay. New species descriptions. This is being done in many, many different ways today. It used to be simply a paragraph, and over the centuries, it's become more sophisticated that we want to have a good illustration, particularly of reproductive parts. That's often important for insects like beetles. This was a beetle. This was my very first part of my very first set of illustrations of new species. And I did this when I was living in the Florence, Italy area. And I worked with Luca Bartolozzi, who is an authority of Lucana beetles, and this was one of his new discoveries. And it was a joy for me to take such a good close look. It's not a very large insect, it's probably a centimeter or less. New descriptions of other taxa, certainly the family level. This is by a dear friend of mine, Carol Hickman, wrote a paper on Croceolidae, which is a new family of these microgastropods, which microsnails, and these are fossils. These are no longer living. So that's still important, especially the paleo world are continually discovering and naming and describing new species. And the illustrations were an important part of this paper to show some of the distinct features of this family. These were also very tiny. So these shells were no more than 5 millimeters high. I don't have a scale bar on these, but they're quite small. And this was another real joy to work with Carol on um, going through the drafts for this and coming up with the finished illustrations. Another important way that illustrations can serve to be a visual communication is through recreation of extinct species from fossil evidence. Richard Thomas, no, I'm sorry, Roger Thomas, maybe he's here in the audience, I don't know, but hi, Roger. Haven't talked in a while. Roger was working on this early gastropod. It's kind of it's very different from our modern gastropods or snails. Very different features to it. The fan-shaped structures on the side are actually setae, which are something that annelids, worms like earthworms, have microscopic structures, and they are believed to have aided this animal in locomotion. He also wrote in this paper about the early torsion. So mollusks have something called torsion, where the organs are rotated 180 degrees with respect to the head. That's one of the distinct features that makes a snail a snail. And he theorized the organ placement. So this drawing also shows the organ placement to show you how small the fossils were. This is what it looks like. So this is a composite drawing taken from many different views and examples of the fossilized remains of this organism, and also very small. If you see, can you see my cursor? This little scale bar is only 2 millimeters.
Ooh.
Pretty small. Back to Carol Hickman again. I've worked with her a lot over the years, and this paper was about recreating the life positions of extinct species. So for a long time, there were alternate views of how this clam animal would be situated under the sand. And it was originally thought to be placed as in figure A. But then she talks about the byssal threads having sand grains on them. Also, and these are also very, very small. And then figure C is what she believes to be the life position, that the animal would anchor itself in the sand with its byssal threads attached to the grains of sand. Another fun project where we're looking at evidence and a lot of trial and error to get the images that she wanted. So this was fun. Behaviors are something else. Now, if you're lucky and you get a photograph of certain marine organism behaviors, great. If you set out cameras and catch it, great. And if you take still photographs, wonderful. But these I was working with a scientist named Roland Anderson. He's no longer with us, but he was lots of fun because he would do dives, scuba dives, and discover some of these animals doing certain things, certain behaviors and interactions with others. And he made some cases for why they do these things and why it matters, especially for other science that comes after it. So these were some of those examples. Another behavior one, Before you even read the first thing, I am sure you can already figure out this has something to do with mating. Yes, you're right. These are cone snails. They're simplistically drawn with the male and the female indicated of each. And yes, they go through this courtship dance of sorts before they copulate. And this was also something that was hard to capture just in a video with all the turning. So sometimes to simplify something in line drawings like this is very helpful in a publication. Okay. Field guides, certainly. So one of my current projects is to have published a field guide of the land snails of northeastern U.S. It's been in various draft stages for over a decade now. The scientist who is leading it, Ken Hotop, sadly passed away. So I'm picking up some of the pieces and trying to get this published. The other illustrator is Kathy Schmidt, a good friend of mine. I'm finishing her illustrations and colorizing some per the publisher's request. But to have good, clear illustrations of snails is important. Some of these in this drawing of these drawings are these are representing very, very tiny species. So these here, where if you see my cursor, very tiny. They're no more than 2 or 3 millimeters high. And this guy here is a Valonia snail, and it's maybe 2 to 3 millimeters at its greatest shell width at maximum. So, gee, I keep getting projects working on really tiny things. Yeah. To switch gears to something much larger, I don't do a lot of this, but my friend and peer through the local chapter of GNSI, Charlotte Tysall, did a beautiful job, I thought, with these. This is something that you can have an at a glance takeaway without even having to read too much. This type of illustration, by the way, is called an infographic. So it has a balanced combination of illustrations as well as text. And probably the most important takeaway is in the upper right is trees growing at 2 different positions. One is in a rocky substrate within soil. The one on the right is growing more comfortably. But look what happens to the rings of this tree. So it has years where it's more sensitive than the well-rooted tree in soil in terms of environmental conditions changing its growth from year to year. But a quick takeaway from a nice, concise illustration. And then there's astronomy, not something I do, but my peer Britt Griswold worked for a long time. I think he still does for NASA, making illustrations of space. Um, and we have, despite having so many beautiful, wonderful photographs of space, to have an illustration that is also an infographic that's explaining the parts and the expansion since the Big Bang can have a lot of information in one image here, and it's pleasing to look at too. What about nanostructures? Well, this is a friend and colleague and peer of mine, Jillian Dittner of the Cornell Lab of Ornithology. She's an illustrator there. And the challenge here was to explain hummingbird iridescence. Not an easy topic because hummingbirds do not have any pigment color. It's all structural color. And what do we mean by that? So she breaks it apart. So look at the feather first. We can imagine the hummingbird feather, which is actually quite small. There's the actual size, and I think that's pretty true on most people's screens. And then she breaks it down to the smaller parts. Barbule is the next thing. And this here being a barbule, and she takes the cross section of the barbule, and over here on the right side of your screen, you're seeing cut cross-section of the barbule, showing the nanostructures inside, which are these kind of hollow tube-like structures. And the color we see bouncing is the light bouncing off of nanostructures, very, very tiny. And the way your angle is from the feather is how you view the color. So you can turn the feather and see it as either yellow or orange or red in this particular picture. But it's an amazingly concise way of explaining something that's very, very difficult to absorb. And believe you me, there's a lot of papers on there that I've read and have had some challenges to understand myself. I've done a version of this. Um, and also want to say public places and murals enhance public places. These are by Jane Kim, an artist whose company is called Inkwell. And a number of years ago, she was a Bartels illustrator, like a fellowship as an illustrator at the Cornell Lab of Ornithology. And her big project was to paint the wall of birds, which you can go into our newly renovated, by the way, visitor center and look at. If you're not close by and you want to see them, you can go visit them online and be able to zoom in to details of her beautiful mural of worldwide representatives of bird families. And I'm going to end this section to say that photography, yes, is important, particularly digital photography of small things. This is from Jeff Thompson of GNSI, who lives in Australia and had his career at Queensland Museum. And so, a lot of you on this call probably know that museums these days have the challenge of digitizing specimens. And what better way that's cheap and inexpensive to digitize specimens is to go through making nice, clear photographs of them. They're also what's called stacked images because they're three-dimensional and you want to capture each detail, even if the specimen is rounded. So Jeff has this technique to do this, and he's done many, many, many of these. So yes, photography is not just relevant, it's better at some things than illustration is, and this is one of them when you're digitizing your collections. Okay. Okay. So the next part of my talk, I'm going to be asking you this question, and it's, can you picture this? So I'm asking you, my viewers, to turn on your mind, read a brief statement or scientific excerpt from a paper, and see whether you can grasp what is being talked about here. So just by words. Can you imagine how proteins interact with the phospholipid bilayer of an animal cell? Some of you might not know what all that means, and that's okay. That is okay because I'm going to show you the picture. So here is the picture of that. This was beautifully rendered, drawn, and then digital illustrated by my friend Elizabeth Morales, another longtime member of the GNSI, and she's currently the president of our GNSI local Finger Lakes chapter. So the phosphobilipid layer this is the wall, the outer layer of an animal cell, right? And it has the lipid part and um, there are lots of structures that are inserted in it that go through it. Some don't go through it, but this shows it oh so beautifully. And I wish I had an image like this when I was studying biology decades ago because they didn't make them nearly as well then. You had to imagine the sentence I just read. right, you couldn't see it as well. So illustrations are quite sophisticated like this, and they're definitely serving as visual communication for concepts that might be hard to understand if you're a student. So thank you, Elizabeth, for the use of your beautiful image. How about this? I don't expect you to read all this. It's past 7 o'clock, and if you're having a cocktail or a mocktail, mites move freely on foot through newly opened flowers within an inflorescence, and they rest in refuges beneath bracts. Okay. Um, have you already had enough of that? Okay, let's just see the picture. This is fascinating to me, and I did not have any correspondence with the scientists who wrote this paper. But did you know that there's tiny little mites called flower mites, and they scamper up the bill of a hummingbird as the hummingbird goes from flower to flower? And they kind of hang out and hang on in the nostril. So I made it a little bit fun and funny. This is the nostril boarding station here. And they hang on and they go along with a hummingbird to the next flower where they scramble down and then they're in the next flower. And they too feed on nectar, in fact. And this is the best way is to hitch a ride and get from one flower to another because it would take a long time if you're a teeny tiny mite like that. So this was just a lot of fun and it was a fun way for me to explain and illustrate what's going on here. All right, next one. Can you picture this? Okay, maybe coffee in the first thing in the morning, but this is hard. I did this project and it's hard for me. Tegument on dorsal rostrum folds into anterolateral auricles. Okay, have you had enough? Okay, I'll just show you the picture. This was an illustration I did of a new species of rotifer. This was discovered by my good friend Aiden Orston, and he found this in his backyard in Maryland. He's been looking at rotifers for decades and studying them, and there's not that many people working on rotifers. They're microscopic animals that dwell in, say, a drop of water that's in your lawn and other places. But they're very curious, alien-like little animals that I really enjoy looking at. And he named it after his wife, Baysoon, which is quite sweet. And just to show you a little more of why this new species description and paper needed both the photos and the illustration is this. Rotifers are hard to see. They are squishy. They turn and move and compress and expand a lot in their as they're living. So the illustration was a way for Iden to clearly depict the internal organs, chosen internal organs of this animal and the features that make it a new species. So I had lots of fun working on this project. How about household dust? Does anybody know what it looks like? We breathe it all the time and we see it and we vacuum it and all that.
Nope.
So wonder no more. Uh, dust. So I made this infographic to show some of the components of your average dusty room. And includes everything from pollen and skin and hair and volcanic ash and cosmic dust and microplastics and pathogens and dust mites. I'm not sure if you feel better after seeing this, but it's easier to picture in your mind than it is to try to imagine or read about it. So this was a fun infographic to do. Back to hummingbirds. A lot of hummingbirds in here because I work for the Lab of Ornithology and we've done hummingbird projects. Okay, tongue morphology. Has anybody watched a hummingbird drinking nectar in their nectar feeder or watched them in flowers? It's very hard to explain what's happening. And it happens very fast because a hummingbird will lap up its tongue and lap up nectar approximately 12 times a second, I think it is, maybe more. So all this is happening. Here's a lot of jargon about it, but here, my colleague at the Lab of Ornithology, Shayna, Muller and I put together this. I did the illustrations, and Shayna had animated it, which is a animation is a great way to visually present something that's complicated. And I'm gonna, as this is going on, I'm gonna read you what's happening. I don't have the original coding in this, but this is just what's on the screen. So hummingbird sticks its tongue out, and it's got these two kind of rolled up structures, and it opens as the tongue goes into the nectar. It's going into the nectar in the flower. Tubes spring open, and then the forked tips unfurl, revealing a row of flaps along the edges. Create suction like an eyedropper, and then as it retracts the tongue, the flaps roll back up. And then it traps the nectar, and here it goes to the second part. It extends its tongue again, and it presses down on the tongue with its beak, squeezing the nectar back and down its throat where it swallows it. Happens incredibly fast. It took us a lot of watching. Yes, we watched videos. There are videos like this, but to have a very clear, clean, simple graphic is what we wanted to present to the masses, because our courses are intended not for scientists. You're a scientist, you can certainly watch them, but for the general public to enjoy and learn something new about birds. So thank you, Shayna, and thank you to the lab for letting us use the little animation here. Okay, can you picture the internal anatomy of a snail? So yes, I've done some dissections, but there is nothing quite as good out there as this. My friend in the Netherlands, Mika Roth, has produced this animated snail. And let me see, I might need to exit out of the slide for a second to retrieve the link for the animation. She's not ready to share the link because it's going through some beta testing now, but I'll show you real quick how this works. Oh my gosh, it's already 7:48. Um, so you can load this model on your computer, and you can turn it all around. How cool is that? But even better, you can look at the individual systems. So did you know that snails have a little beating heart? Well, let's see it. So here's the vascular system. That is some of,
You need to go back to sharing your slides.
oh no. All right, let me do that again. Thanks for letting me know. Snail app, please. Okay, thank you. All right. So this is Mika's app. So you can use your mouse or cursor to control the movement and the angles. We're looking right now at the vascular system of the snail. So the heart of the snail is over here. It has this two-chambered heart. And this is the lung, this side. The vascular part of the lung that brings oxygen through its blood system. And you can look at other things like the nervous system, the digestive system, which is weirdly like our own, and the nervous system. Very fun app. So it's going through some beta testing now, and it will be available in time. So keep up with Mika Roth. I'm going to go back to the PowerPoint. Let's see, share this PowerPoint. Okay, I'll get us back there, I promise.
You're back.
Yeah, great. All righty. All right, good. So the next, I'm going to conclude my talk with something that is personal to me. I'm only speaking for myself as an illustrator, but I don't doubt that a lot of my friends and peers in the illustration world might have some similar feelings, but I'm going to go through some of my slides and read you my account of what it feels like to be an illustrator. So, okay. So to be an illustrator, an artist, I feel incessantly curious about all parts of nature. I ask questions every time I see a snail or a spider or a willow tree or an osprey. Uh, and I ask to myself, what is it doing? Where is it going? How does it do this? Why is it doing this? And as I do this, I'm experiencing the shapes and the forms, the textures, the rhythms of nature. I try to understand its perfections, its imperfections, its subtle nuances. I feel incredibly humbled by nature, and I always feel that I can never produce anything that's as good as what's already there. Then I try to return to my purpose as a visual science communicator. I'm just simply serving as the conduit between nature and the learner. So I absorb as much as I can by looking and studying it, and I let it get fixed into my brain. And then there's the hard work. There's a lot of hard work and years of learned techniques to apply, lots of conversations with the scientist or the person who's commissions the work. And then there's lots of mistakes and sketches and tries and retries. And I do this in isolation and it's laborious. And then I try to think about the person on the other side who's going to receive and look at what I am doing. And as I think about that person, I hope that they will experience what I've drawn with clarity and a newfound appreciation if I have done my job well, that they will understand it better, appreciate this part of nature, and they will care about it more. That's my presentation for you tonight.
Thank you. That was lovely. And I'm going to put a link in the chat to let people know where you can find the video. But it's time for questions. So I'll open it up for questions. We've got a pretty good crowd. So if you use the React button, which I see many of you know how to do, which is great, to raise your hand, then you'll raise to the top of the list of folks. So um, go ahead and ask some questions. And there's the link where you'll find.
I, I have
Go
a
ahead.
question.
Go ahead.
Can I speak?
Yes, go ahead, Gustavo.
Well, the bug in the nostril in or near to the beak of hummingbird is like a parasite or is so different?
Oh, it's so different. So the hummingbird's bill is in this case that I had studied and drawn, it's not a parasite. It's a type of symbiotic behavior called foracy. And the closest example that you are probably more than familiar with is, have you ever gone for a hike and gotten a bunch of sticky seeds attached to your pants and socks? That is foracy. Those seeds are just hitching a ride on you. That's it. That's all they're doing. They're not a parasite. They're just saying, hey, I mean, they're not just saying anything because they don't have brains, but they're seeds and they can get carried around on your parent, your genes, or if you're a deer or rabbit, they'll get stuck in your fur, or if you're a dog, and then they'll fall off later to be where they are going to survive and thrive in hopefully. So the hummingbirds' mites are just simply hitching a ride to go from flower to flower.
Just reading the comments in the chat to see if there are questions, or is mostly just praise.
Thank you.
Are there more questions?
Um,
I, I have one.
go ahead.
Can you tell us you touched on it a tiny bit, but can you tell us a little bit more about how you became a scientific illustrator or visual communicator?
Sure, sure. I was in my fourth year of undergrad studies as a biologist at University of Georgia, and I realized that there's a course in science illustration. It's being taught by Lloyd Logan, and he was one of the original founders of the Guild of Natural Science Illustrators group, I believe. And he was just an excellent teacher. He encouraged me. He encouraged everybody. I was really into it because it just made so much sense. I had not known previously that this was a thing. This is a valid path to pursue. So yes, I got my degree in biology, but I had that under my belt, and then I joined GNSI and then went on to continue my learning through everywhere I go, through job positions and talking with scientists and practicing and having job opportunities.
So,
Always loved to draw as a child. Yeah, I couldn't get that out of my system. As soon as I could hold a crayon, I was drawing everything. And I love to explore nature in the backyard or anywhere to find tiny snails, to find the insects, to look at the leaves and seed pods. That was all. It was just, I wasn't a social child so much. I really just wanted to spend time in nature and draw.
and now there is a question in the chat. How will your next book be showcased? What audiences?
So the field guides are two kind of sub audiences of those interested in snails. One is one of the publishers is the type that makes the fold out laminated guide that are very popular, very easy to take into the field, doesn't require time to page through or anything. So you can um, rather quickly identify in the field what you're seeing, one of the snails, different views of that being drawn. The other book is a more in-depth one. That was the original plan, I believe, before the lead author passed away. And that's also, I'm getting a lot of encouragement from a publisher to get everything together and finish the illustrations and get that going. Yeah.
Other questions?
I might, if there's not, I left one more slide here because what I didn't talk about in this presentation was the process itself and the media that I use. Um, we talk about illustration media in two broad ways, traditional and digital. Traditional being everything from pencil, graphite, to charcoal, to ink, to paint. Okay, and then digital is Adobe Illustrator and Photoshop, Procreate, and these other computer programs that we can illustrate. Sometimes we use both. Let me see if I can get to my next slide. Okay. Um, so these, I used to do everything in ink, and then come the digital age, I would start out with pencil sketches. In fact, yes, always, but then trace them over with digital line. And here is the reason. This is a conversation between me and Iden Orston about the details of his new species of rotifer. And these were his notes on top of my early versions of the illustration, the top two. He's asking me to make some changes. And then I made the changes. Of course, there were still more changes to make. I think this is a very abbreviated version of it, actually, because one goes through a lot of versions before they hit it right. And you want your scientist to be completely happy with the illustration that you've made, because it's got to stand up in the scientific community and be reviewed and be considered a valid new species. So this was part of the process of that.
That's very cool. And I think William Bryce had a question.
I'm
Go
just
ahead.
wondering, do you work with vertebrates other than birds?
Do I work with vertebrates other than birds? Not a whole lot. I kind of do for fun. I am very interested in birds, and it's part of the reason I work at the Lab of Ornithology. But most of my background is in invertebrate zoology. My graduate school work is in invertebrate zoology, specializing on land snails. And I just seem, I don't know. I think I've always been this way. I tend to root for the lesser charismatic, lesser appreciated animal life in the world. And so many more people know what an elephant is than a rotifer. And I think rotifers are beautiful, and I think snails are beautiful and interesting. And I think that they deserve, um, I think they deserve people's attention. It's a hard case to make sometimes, but partly through illustrating them and showing off how amazing they are, that what incredible structures they have at such a tiny scale is interesting to me.
Cool. And a couple more questions have appeared in the chat. Is AI changing your practice?
AI changing my practice? Yeah. Yeah, it's concerning because we are all as illustrators up against it. And the very sad thing that I'm starting to find in my work life is that when we're looking for images, we have the Macaulay Library, which is wonderful, but there are a lot of AI images of birds and birds' nests and things like that out there that are just plain incorrect. And it hurts me to see them, to think that people are believing them and think that they know what something looks like or how it behaves or its structure through an AI image. So I find it very discouraging. Um, uh, that's how I feel about it right now. I know AI can do wonderful things. It can solve problems and faster, but I discourage
I,
its use of trying to make any kind of biological organism through it.
okay, and there's a couple more questions. The last one is a follow-up to that one. So that is, are you actively involved in combating combating that misinformation?
Well, not directly. What I put out in my work, both at the Lab of Ornithology and in my freelance work and other side pursuits, is the truth. And science is upheld for a reason. It's based on facts and observations, and it's vetted through other scientists and peer reviewers. So by sticking to that and explaining to people that that is the truth, I'm not actively, I guess, no, I'm not really actively doing any anti-AI advocacy, but I'm thinking that's coming along for anyone who considers themselves a visual science communicator, with that being a threat in the future. I don't have time. I barely have time to do all the jobs that I'm doing right now. But yeah.
And the Sarah May asked that question and added a follow-up to it that says, ask, as a active science communicator teaching the public, I can tell you that does not work. Nothing against you or anyone else, but it is the evidence-based reality. Heavy sigh. Um, and then another question, do most scientific illustrators have a focus? If so, how specific? Does a scientific illustrator come about, or does a scientific illustrator come about their specialty? I think there's a how missing there.
Yeah, yeah, absolutely. One of the most fun things in my life is to go to a Guild of Natural Science Illustrators conference. They now happen every other year. Unfortunately, I wasn't able to make it to this one. And there you meet your people. and everybody's interested in different things. So you meet people who are interested in pollination and they do wonderful illustrations of that. And you meet people who are interested in marine mammals and dinosaurs and tiny seeds and fungal spores. I mean, yes, you get into your specialized areas. Mine is snails. It doesn't mean I don't like to dabble in other areas. Rotifers I quite like as well, and I'm interested in fossil mollusks. But yeah, we get into our specialized areas and find our niches, and then you get known for that. And that, the more of that you do, people will know which of these illustrators to ask for which projects.
And there's a more direct question or something. Can I keep in touch with you?
That's
So,
very nice. Uh, I will do my best. All my friends who are on this call probably know I've just kind of fallen off the edge of the planet this year just because of a lot of personal challenges. But yeah, you can see my website. It's just marlacoppolino.com. Just my name, no dots or anything, marlacoppolino.com. And there you can find space to contact me.
and a couple more follow-ups from Sarah May. I would encourage you to use your expertise in recognizing misinformation and pass it along to those of us working against disinformation. We need people like you. And she added a website there. And
Is it in the chat?
that is in the chat, yes.
Can we save the chat? That might be helpful.
And then there's another question. What would a very good day look like in a day and life in your a day in the life in your profession?
Well, well, at my day job, I do a lot of things there. I'm everything from script writing for the narrations for our videos to making decisions on what goes on our course pages and all of that. But a very good day would look like one of the days where I'm working on illustrations and talking to my colleagues at the lab about that and having meetings about what's going to work the best and trying to use our creative side and our science side together to present a difficult to understand topic. That's just so much fun to me. It's like puzzles. It's like, I also like crossword puzzles and things like that. But the visual illustration puzzle to me is super fun. And then the second part of that is if I have time when I get home from work to pursue some of my own, to keep working on some of my own illustrations. It doesn't happen every day. I try to have it happen on the weekends, or and to keep sketching, field sketching. That's an important part of my life. I love. So at the lab, some of us go out during the lunch hour with our little field painting sets and we just draw and paint around the pond, and that's great fun. So yeah, a day in the life would be that I'm doing a project at work, that I get the lunch break to go do some field sketching, and that I also get to draw when I get home. Good
Other
question.
and lots of resources about misinformation being shared in the chat there too.
Thank you. I'll try to capture that chat.
Other questions?
Well, I have another question. Uh, Dr. Coppolino, can I keep in touch with email or with other media
Keep in touch with me by email or what? I didn't quite understand.
or other media? Sorry.
Other media? Uh,
Yes.
Well, maybe others. I encourage everybody. First of all, yes, you could go to my website and I'm accessible there. But also go to the Guild of Natural Science Illustrators website, and you will be graced with a world of what illustration is about, what visual communication is about. And you can see lots of work of members. There's galleries, so you can see what a lot of us do.
Well, but how can I find your website?
Oh, marlacoppolino.com.
Put it in the chat.
Yeah, let me put that in the chat, and that would might help. So it's just put this in your search engine. Marla, whoops, .com. Pretty easy.
There it is. Other questions? Yeah. Here, I'll put a couple more links in there and say very nice opening session for the fall. Very good to see a lot of our regulars again. I've missed you all, so welcome back, and we'll see you in a couple of weeks. And thanks one and all, and thanks especially to Marla for a great presentation and discussion.
Thank you, everybody. This was fun, and I appreciate your time to listen this evening. Thank you, and thank you, Don, again.
Thanks all. Have a great night.
Try to preserve the chat, save chat.
Oh, and I will let you save the chat before I hit that end button.
It usually goes to some mysterious folder.
Right.
All right, hopefully that worked. All
Yeah,
right.
if you can't find it, I can send it to you because it
Thank you.
automatically.
Okay, sounds good. All right. Thank you again. Thanks, everybody. Have a good night.
Thanks all. And again, this will be up. This is already up on our YouTube live